is a wonderful opportunity for me, because over the years, you're lucky to work with a few extraordinary people, and without a doubt, Shan Huang is one of those people. And she's not only extraordinary, but an incredibly nice person in every way. But before I go on to introduce Shan Huang, Felix asked me to say a few words about the Integrative Center for Learning and Memory, because this is the Integrative Center for Learning and Memory lecture. Or our center for short is the ICLM Center. Well, our center has been around for at least 20 years. It became officially a center more recently, but the faculty in our center have been meeting and collaborating for at least 25 years. We have about 46 faculty in our center from 9 different departments in the medical school and in the college. Our center, as you may imagine, is a collaborative hub for faculty studying plasticity, memory, and associated behaviors. Over the years, the center has been the hub for training grants and center grants that bring groups of faculty together. We also have a long-running, very active journal club, which I invite you to join. It's a lot of fun. It meets on Friday at 9:30 a.m. in the second floor Ganda building. We have also had yearly meetings that bring together faculty in our center, of course, and in other learning and memory centers worldwide, with an emphasis on Southern California. So we have become a hub, really, for a learning and memory meeting that takes place every year. We also have a number of programs that may be of interest, to you, including workshops. So we run workshops, periodically. The last one that we have run was on head-mounted forensics microscopes that were developed here at UCLA. We run a couple of undergraduate and graduate courses. We have a lecture series for distinguished, contributors from our students and postdocs. So, really wonderful findings within our center. We have this lecture series that highlights them. We have a program that faculty can take advantage to review grants and papers before they are officially submitted. And this has been actively used by many of our faculty. We have a yearly prize awarded, to graduate students and postdocs in our centers that have made extraordinary contributions. We have a distinguished lecture series, etc., etc., etc. Anyway, if you're doing things related to plasticity and memory and associated behaviors, I highly recommend that you join us. It's a lot of fun. The activities of the center are managed by a council that includes myself, David Glanzman, Dean Bonomano, Kate Wassam, Michael Fenslau, and Michelle Basso. So if you have any questions about the center or what's going on in the center, actually we have a core facility, a behavioral core facility. There's a lot going on. We have a website that includes all of this. If you have questions, you can contact any one of us. But now to the real activity that we are all here for, which is Dr. Shan Huang's talk. Let me introduce her briefly. Dr. Huang did her undergraduate at Nankai University in China, where she studied microbiology. She actually did undergraduate research. During this time, she won a number of prestigious awards and fellowships. And it was one of these fellowships, which is for exceptional undergraduates, that allows them to go and study abroad, that brought her to UCLA. And lucky me for that, because after doing an internship in our lab, I managed to convince her to join us and do her graduate studies with us. And she did extremely well. She published a number of papers in our lab, but I would only mention one. Together with Adam Frank, Shan used in vivo 2-photon microscopy and discovered that dendrites have hot spots for spine addition during learning. And that spine turnover before learning in these hot spots predicts future learning and memory performance in animals. Amazingly, she also discovered a genetic manipulation that enhances the activity of these hotspots before learning, and therefore enhances learning and memory and spine clustering. Importantly, the stability of these spine changes can predict, within these hotspots, can predict memory stability. Really a beautiful series of findings that wonderful studies that she carried out in the lab. Then she moved on to Weizhe Huang's new lab at UCLA, where she has been studying circuit mechanisms of social behavior, the topic of our lecture today. Without further ado, it's my enormous pleasure to welcome Shan, and I'm really looking forward to this lecture. All right, Shan, the time is yours.
Well, thank you so much. Thank you so much, Alcino, for the wonderful introduction. And thanks so much for Felix for providing this opportunity. And it's my great honor to be here and represent the Integrative Center for Learning Memory and share with you our recent exciting work. Okay, let me start sharing my screen. Can everybody see my screen? Okay. Okay, today is my great honor to share with you our recent exciting work about how sex information of a social partner is encoded or represented in the prefrontal cortex, and how those sex representations shape animals' behavior. Social behavior is essential for animals' health, survival, and reproduction. Many types of social behavior are evolutionarily conserved, so they exhibit similar features across species, from invertebrate to human. Such behavior include social affiliation, social dominance, mating or parenting, etc. Deficits in social behavior are associated with many neuropsychiatric disorders, such as autism spectrum disorder and schizophrenia. So it is important to understand the neuromechanism underlying social behavior and develop novel treatments for disorders with social dysfunction. What is unique about social interactions is that they involve active detection of a series of sensory cues and integrate those cues from the other individuals or the environment with their own internal states to generate a behavioral response. Looks complicated, right? But it actually happens every day in life. So take my 8-month-old baby as an example. When he was frustrated, he cried. And when I heard his cry, as a parent, I worry about him. So I call his name and sing his favorite song. He heard my voice and he felt less frustrated. So he stopped crying and smiled at me. So interactions are shaped by this dynamic and mutual feedback between participants in real time. As social interactions involve more than one individual, an open question is how our brain recognizes cues from other person and transform those sensory cues into adaptive behaviors. Like, how does my baby recognize my voice and start to smile? A lot is going on inside this black box. The recent 2 studies in our lab shed light on this question. The first story I'm going to share with you identified a subset of neurons that specifically respond to the behavior of their social partners. Those neurons made a great contribution to the brain coupling within the pair, which can drive future behavior interactions. And the second story is about how our brain encode sex-specific cues and shape sex preference behavior. I'm going to introduce you the first story briefly. It's published, and focus on more details of the second study. Let me make out the pointer. Okay. In both studies, the main approach we applied is the miniscope developed in UCLA, initiated by Payman, Alcino, Ball, and Daniel. I believe a lot of you are already very familiar with this approach. We implanted green lens into the medial prefrontal cortex, or MPFC, which has been shown to be important for social cognition and social decision-making. Miniscope captures neural activity indicated by calcium fluorescence and allows us to record from hundreds of neurons per animal with single cell resolution. And the animal is freely behaving while we are performing the recording. For the first study, we adopted a competitive social context, which is called tube assay. Two mice are set to meet each other in the middle of a narrow tube, which allows only one mouse to pass at a time. A dominant one may push the subordinate one out of the tube, or the subordinate will spontaneously retreat to give way to dominance. In the tube assay, animals mainly display two types of behavior, which is push and retreat. We simultaneously record neural activities from two animals while they were interacting in the tube. In this way, we can get neural responses of two brains at a time and see how neural properties across brains will affect their behavior and relationships. Surprisingly, we found that their neural activity was synchronized. And this synchronization was significantly diminished if the two animals were physically separated in the tube, suggesting that synchronized neural activity is not due to the similar sensory impulse from a shared environment, but actually depends on ongoing interaction between the pair. We also test the animal in a non-competitive context, like in the open arena. We still observe the synchronized activity across brains, and suggesting that the interbrain synchrony is a general phenomenon during social interactions. Although the interbrain synchrony was previously observed in human studies, little was known about the neural mechanism underneath this phenomenon. So to explore what makes the two brains synchronized, we extracted calcium traces from hundreds of neurons per animal and analyzed how those neurons responded to their own behavior or the behavior of their opponents. Surprisingly, we found quite a substantial fraction of cells in our brain that specifically responded to the behavior of their opponents. And next, we want to see how synchrony is affected by those neurons encoding self-behavior or opponent behaviors. If you remove either self-encoding neuron or opponent-encoding neurons from the dataset, the interbrain synchrony dramatically reduced in both cases. In contrast, if you remove other cells, they don't have an effect on synchrony, suggesting that neural activities responding to self and opponent behaviors are essential for the synchronized brain activity between the pair. These cells allows each brain to encode a set of common features, that is the behavior of both animals, such that activity across separate brains becomes synchronized. Next, we want to ask a more important question. What is the behavioral relevance of interbrain synchrony, and what can we learn from that? We found that the likelihood that one animal would have a reaction in response to the other animal's behavior can be predicted by the degree of synchrony right before the interaction starts. For example, animal A pushes on animal B. If the synchrony is high before animal A pushes, it's highly likely that animal B will have a retreat or pushback as a response. If the synchrony is low, animal B may not react to animal A's push. In addition, we show that interbrain synchrony can also predict the dominance relationship of the interacting pair. Animal with equal dominance status will have a lower synchrony, while those with very different dominant status, like one is very dominant, the other is very subordinate, then their brains tend to be more synchronized with each other. Taken together, the above study shows that the behavior of others can be recognized by neurons in our brain as sensory inputs. Those opponent encoding neurons and self-encoding neurons together influence the interbrain synchrony between animals. Interbrain synchrony not only arises during social interaction, but reflect the dominance relationship between animals. In addition, it may actually influence the behavior decision of one animal in response to the other. Similar like whether I will have a behavior reaction in response to my baby's cry. In addition to recognize and predict behavior of others, an innate function of our brain is to recognize the sex of others. My next story is about how brain recognize sex and shape sex-related behavior decisions. We just talked about social interactions in a competitive context, where animals display dominance behavior. Dominance behavior is usually observed between animals with the same sex. Animals may fight with each other of the same sex of territory for territory, food, or mating partners. On the other hand, many types of social behavior have a preference for partners with the opposite sex, such as mating behavior. So you can see that animals display very different types of behavior, which is dependent on the sex of the other animal. Therefore, being able to recognize sex of the other animals and distinguish one from the other is critical for animals to make behavioral decisions, which are important for their survival, well-being, and reproductive success. So how is sex information processed in a rodent brain? Those chemosensory cues released by other individuals are processed in two separate olfactory systems, the main olfactory system or the vernal-nasal system. Odorants or pheromones released by other animals of different sexes are detected by a specific populations of sensory neurons in the two systems, where they activate distinct receptors and send distinct inputs to the main or accessory olfactory bulb. Then they converge onto a region called medial amygdala, where sex-specific neural response start to show. And then they further diverge into different hypothalamus centers to trigger sex-specific behaviors, such as aggression, mating, or parenting. Lots of literature about sex cue processing are within these subcortical regions, leading to a long-standing view that processing of sex information depends exclusively on subcortical circuits. However, little is known about sex representations beyond those regions. Although sex-specific behaviors are evolutionarily conserved, but the processing of sex cues, especially in the lower order neurons, may vary across species. For example, as a human, we don't rely on chemosensory cue to recognize sex of another person. Instead, we mostly recognize sex through visual stimuli, such as face and body features. Like the diagram I showed you earlier, such many complex social cues from other individuals, from the environment, as well as our internal states must be integrated somewhere with a dynamic coding system to accommodate outside changes and make adaptive behavior decisions, such as dominance or sexual behavior. Compared to the subcortical circuits, cortical circuits are more plastic and adjustable, since they may be suitable for such a role. In addition, it has been shown by works from our lab and also many other groups that MPFC, medial prefrontal cortex, in particular, is involved in modulating sex-specific behaviors. All of this motivates us to look beyond subcortical regions and investigate how sex is encoded in cortical regions. To give you a brief overview of the data I'm going to present, I will first introduce our findings about how male and female social cues are encoded, or here we call represented in the MPFC. And next, I will provide evidence to show how activity of those sex-encoding cells in turn influence animals' behavior while interacting with other male or female animals. The first question we wanted to ask is whether MPFC neurons encode male or female specific information. During miniscope imaging, we presented each subject mouse with multiple novel males, novel females, and a toy mouse in random order. The subject mouse can freely explore each novel conspecific for about 20 seconds. After the whole session was done, we extracted the single cell activities and aligned them with the behavior events to classify whether a cell is selectively responsive to a given stimulus, and classify cells into categories of female responsive, male responsive, toy responsive, or unresponsive to any stimulus. Here shows some example cells. The top row shows a cell which has a mixed response to both male and female. This category only occupies a small fraction of the population we have imaged, and the more cells have a selective response to either male or female investigation. We cluster single cells into groups. For example, male excited neurons are those increased activity in response to a male interaction, and male suppressed neurons are those decreased activity during male interaction. The same for the female excited or female suppressed cells. It's clear to see that the excited cells have a time-locked increase of activity at the onset of investigation of the given sex, indicated by this small triangle. But the same cells do not have much activity during investigation of the other sex. These results visually confirm that the sex-encoding cells we have identified are selectively tuned to only one sex. And next, we want to compare the strengths of encoding of male versus female cues. In female animals specifically, we observed an equal fraction of neurons that were excited by a male or female social partner. Interestingly, male animals have a larger fraction of neurons that are excited by female investigation. This finding is quite intriguing as it suggests two things. Number one, the encoding of sex information differed between males and females. Number two, male animals have a larger representation of female information than male information. In the following analysis, we focus on male animals to gain deeper insight into this asymmetric encoding of sex information, why males have a stronger encoding of female social partners. And next, we want to take a look at how sex is represented at the population level, which should give us a more stable and reliable response than individual neurons. We first simplified the data using principal component analysis, or PCA, which is good at transforming a large set of variables into a smaller one that still contains most of the information. Using PCA, population activity from hundreds of neurons were transformed onto this three-dimensional space defined by the top three principal components, which best explain the data variance. Take this animal as an example. Thin blue lines shows the population response during each investigation with a male animal. The thin red lines are those during each investigation with a female animal. Thick lines are average population dynamics across many investigation events. And this showed a clear separation of population responses during male versus female interaction, suggesting that the population activity can distinguish sex information. In addition, we found that male animal showed a higher amplitude of population response during female investigation compared to male investigation, which is consistent with the previous finding that male animals have a larger fraction of neurons that encoding female information. Next, we want to test whether the sex representation is general enough to decode sex across time and across different novel social partners. Here, we applied a machine learning algorithm to train a sex decoder that can predict the sex of a novel animal. And this prediction is solely based on animals' neural response. To train the decoder, we feed the decoder with data that were randomly picked from any time of the experiments and use the remaining data to test the decoder performance. If the brain responds differently to male and female cues, the decoder should be able to detect that difference and use that to predict the sex of a novel animal. Indeed, we found that the decoder performance, which is how accurate the decoder can predict the sex of a novel animal, is much higher than chance levels, indicating that specific sex triggers unique and consistent response pattern in the MPFC, so that male and female events can be well separated. This result again support the notion that population activity in the MPFC can well distinguish different sexes. The sex encoding pattern at the population level is stable across time and general across different identities of social partners. Taken together, both single neuron and population of neurons show selective response to male and female cues. Population response pattern of male and female cues are separate in MPFC. And male animal, in particular, form a stronger encoding of female cues than male cues. Similar as other cortical regions, MPFC consists of different neuronal subtypes with both excitatory neuron and inhibitory neuron. We wanted to gain deeper insight into how sex representations are distributed across different cell types, whether specific neuronal populations are preferentially recruited to encode sex information. To address this question, we injected AV virus expressing GCAMP and driven by a campus2 promoter to specifically image excitatory neuron, while expressing GCAMP in GABAergic cells to image inhibitory neurons. We found that both populations contain cells encoding social stimuli. Remember, I just showed that male animal had a larger fraction of female cells. And here again, in both populations, the fraction, we see the same trend that there are more female encoding cells in male animal across populations, which is consistent with our previous finding. They indicate that the bias in encoding of female cues is preserved across both excitatory and inhibitory populations. Interestingly, inhibitory neurons have a higher fraction of social responsive cells compared to excitatory neurons. And this enrichment of encoding is true for both male and female cues, as inhibitory neurons are more likely to be excited by either male or female animal. We again constructed machine learning decoders and found that both excitatory and inhibitory subpopulations could well predict the sex of a novel animal, with inhibitory neurons have a better performance when using the same number of neurons to train the decoder. This suggests that inhibitory neurons contain more sex information than excitatory neurons, such that male and female responses are more separable in inhibitory populations. So far, this data demonstrate that sex information is distributed across excitatory and inhibitory neurons, while more enriched in inhibitory neurons. And importantly, both subpopulations exhibit a stronger representation of female cues compared to male cues, suggesting that stronger encoding of female is preserved across both excitatory and inhibitory cell types. A main difference of cortical and deep subcortical region is that cortical circuits is thought to be more plastic, so are more likely to undergo adaptive changes to serve the goal. To test whether encoding of sex information undergo such plastic changes over time, we adopt a two-chamber assay as an alternative social context. In our previous assay, we alternate different intruders, and subject mice were allowed to explore each intruder in a given or constrained time window. While in the two-chamber assay, they have the options to free explore either male or female at his own will without disturbance. In this way, it allows us to track the activity of sex-responsive cell over time. we first confirm that sex encoding is stable across these two different contexts. So sex cells have consistent response in both contexts. The heat map shows that male responsive cells selectively increase activity while exploring the male social cup, and female responsive cells were excited near the female animal cup. Next, we tracked activity of those sex-responsive cells over time and test how their response get modified as they spend more time investigating male and female social cues. Interestingly, we found that in cells that responding to social stimuli, the difference of activity between male and female investigation become larger and larger over the course of the experiment. This increase in difference is mainly driven by the reduced response to the opposite sex. Let me explain this data using an example. You can think I am a female encoding neuron. At the beginning of the experiment, I have a stronger response to female, but I will also have a weak response to male because I'm a little confused as this is my first time seeing a male and female in the same environment. But as I spend more time with each of them, I'm experienced. I can better tell the difference between a male and a female. So I sharpen my response to female by reducing my response to male. In this way, the mean response of male and female stimuli becomes more diverged as the animal getting more experience of the two sexes. However, this was not true for cells that responded to non-social stimuli, such as plastic toys, suggesting that sex-encoding cells are tuned to better distinguish male and female cues as they gain more social experience with both stimuli. Another evidence is that the population response decoder that trained using the data from the second half of the experiment had a better performance in distinguishing the two sexes than using the decoder trained by the first half of the experiment. But it didn't show the same enhancement in decoding non-social objects, suggesting that enhancement depends specifically on experience with social stimuli. Take a summary so far. The male and female representation is distinguishable at both single cell and population level, and they become even more separated as animals gain more experience of the male and female cues. These results suggest that neural circuits associated with sex encoding undergo such dynamic changes, which modulate their response strengths to different sexes, since the brain can better distinguish one sex from the other. After identifying how sex information is encoded in the MPFC, here comes the question: what is the behavior relevance of those sex representation? Why should we care about those sex representation? So in theory, the sex representation should play a role in driving sex-specific behavior. In many results that I just showed, there is a consistent trend of stronger encoding of female cues in males. The question is whether the stronger female encoding in males has anything to do with their behaviors. So we looked back experimental videos and wanted to see whether animals have a preference for partners with a specific sex. We measured the time that animals spent with each social partner and calculated a sex preference score, with positive number meaning a preference for interacting with males, and the negative number meaning a preference for females. We observed a group-level bias toward interacting with females, raising the possibility that the female encoding bias may be linked to this female behavioral bias in male animal. Although they show a female preference at average, individual males displayed a wide range of sex preference scores. Some males exhibit mild or strong female preference, but a substantial fraction of males show a preference for interacting with male animal. Previous studies have only looked into this group level toward interaction with opposite sex animal. However, little was known about why some animals prefer to interact with those of the same sex, or where this individual variability of sex preference comes from. So in light of this, we were thrilled to test the idea that whether individual sex preference is linked to its asymmetric encoding of male and female cues in the MPFC. We first group individual animals into male preferring and female preferring group based on their sex preference scores. We measured the population response amplitude during each male and female investigation as the distance between event-triggered population vector and the baseline activity, and found that the female preferring animals showed a much stronger response during interaction with a female social partner than with male partners. In striking contrast, male preferring animals showed a stronger population response during interaction with male animal. This result suggests that the relative activity of male versus female evoked response can predict the sex preference behavior. This well explains the individual variability in sex preference. As I showed previously, we found consistent trend of stronger female encoding in males, which is linked to the overall female preference in male animals. But individual animals showed a wide range of sex preference. We found that animals with a stronger response to female cues, indicated by this longer distance from baseline activity, may indicate a preference for females, while animals with a stronger male response may like spending time with their male friends. So far, I have introduced our findings about how sex information is encoded in the MPFC at both single cell and population level. And we found that behavioral preference of male or female is correlated with the relative strengths of sex representations. To test whether there's a causal link between cortical sex representations and sex preference behavior, we would like to manipulate the activity of sex-encoding cells and see whether that can influence animals' behavior choice of interacting with male or females. We have shown that sex-encoding neurons are distributed across both excitatory and inhibitory neurons. Previous literature also showed that the sex representations are not mapped onto distinct cell types with a simple biomarker. So it has been difficult to specifically manipulate those sex-encoding cells, such that there is a critical gap in literatures about how neural representations of sex affect animals' behavior. Therefore, an activity-dependent labeling strategy is necessary in this scenario, which can specifically label and manipulate those neurons based on their natural response to different sexes. And the particular construct we use is driven by a synthetic promoter called ESER. Here, we adopted the ESER activity-dependent labeling coupled with optogenetic stimulation by injecting a combination of virus. So the first virus is the activity-dependent Cre virus, and the second is Cre-dependent channel-rhodopsin virus. So a social stimulus, in this case, a male or female intruder, drives the expression of Cre-ER, specifically in neurons that are active at the moment. Cre-ER stand by in cytoplasm and will only become active until a tamoxifen is injected. After being activated by tamoxifen, the Cre will come into the nucleus and initiate the expression of channel-rhodopsin. After a few weeks, when enough channel-rhodopsin is accumulated in cell, one can use optogenetics to manipulate the cells that were specifically activated by the social stimulus. This diagram shows how we carry out the activity-dependent optogenetics. Each subject animal was given 1 hour to freely interact with a novel male or female animal in the home cage. After the 1 hour exposure is done, we systematically inject tamoxifen to activate Cre recombinase. A few weeks later, we test the subject animal in 2-chamber assay where we shine blue light in the middle of experiments to stimulate those sex-encoding ensembles and see whether that makes any changes on their sex preference behavior. The control group was not given any social stimulus during the 1 hour exposure time, but they were otherwise handled in the same way as the experimental groups. We used optogenetics to stimulate neurons that were naturally activated by female cues. Stimulation of female activated cells in the middle 10 minutes of the experiments resulted in acute change in sex preference toward female animal. This was driven by an increase in the time spent investigating the female partner, as the male investigation time was not affected. In contrast, the stimulation of male activated neurons dramatically reversed an overall female preference to male preference, with an acute increase in time spent interacting with the male partner. You can see that after stimulation, they go back to the female preference. This suggests that sex preference behavior is modulated by the activity of the native neural representations of male and female cues. This was further validated by the control group, where the stimulation of neurons were activated during a home cage exposure without male or female stimuli had no effect on preference behavior. Together, these results demonstrate a functional role for cortical representation of sex in shaping sex preference behaviors. Let's take a summary. I have shown that different sets of neurons in the MPFC respond selectively to male and female cues. As a population, they exhibit distinct response patterns while interacting with male versus female animals. Those sex-responsive cells are distributed across excitatory and inhibitory neuronal populations in the MPFC, but more enriched in inhibitory neurons. The neural circuits encoding male and female information undergo dynamic changes so that neural response to male and female become more selective as animals gain more social experience. Importantly, we found a consistent trend of stronger female encoding in male animals. They have a larger fraction of female responsive cells. They elicit a stronger population response to female, and this asymmetric encoding happens in both excitatory and inhibitory neurons. We found this stronger encoding of female cue is linked to their overall behavioral preference for female animals. So next, we measured the behavioral sex preference for individual animal and found that the sex preference can be predicted by the relative strengths of encoding of male or female cues. So male preferring animals show a stronger encoding of male cues, while female preferring animals have a stronger encoding of female cues. Then we stimulate those native sex representations using optogenetics. They directly modulate animal sex preference behavior. Sometimes it can even reverse the original preference to the other sex. And this demonstrates a causal role for activity of social representations in shaping social behaviors. This finding together demonstrate for the first time that sex information is represented in a cortical region. We show that sex-specific tuning gets sharpened quickly with social experience. This means that sex encoding in cortical circuits is a dynamic process at a much faster timescale than the subcortical circuits. This dynamic coding system allows for more adaptive behavior decisions to accommodate outside and internal changes. Taken together, the above two studies both provide insights into how information from the other individual is recognized and processed in our brain and affect our behavior response in real time. Those information from another individual can be as dynamic as a behavior or voice, or can be a fixed feature such as sex and age. Our brain have specific sets of neurons that encode those information from the other person, and they elicit distinct activity patterns for us to distinguish one from the other, like distinguish male from female, push behavior from retreat behavior. And the relative strengths of the encoded information may influence our innate behavioral preference. And also this encoding system is dynamic and adjustable, so they can be changed from time to time for us to make adaptive decisions. At the end, I want to highlight my gratefulness to these two gentlemen. Lyle Kingsbury is a graduate student in the lab. He's the other co-first author of both studies. Lyle has made a great contribution to these two projects. Collaborating with him is a delightful experience and very productive. Also, I really want to express my gratefulness to my PI, Weizhe, who has provided me with tremendous support and guidance on every aspect of scientific training and also postdoc career. His scientific rigor and vision not only direct this work, but also inspire me in the long run. Lots of thanks to Lyle and Weizhe. I also want to express my special thanks to Tara, Letizia, and Rongfeng in the lab, who also make great important contribution to the experiments. Many thanks to the collaborator Li Ye from Scripps Research for support on eSARE activity-dependent labeling. And also many thanks for Peiman, Daniel, and Pingping for the generous technical support on miniscope imaging. Thanks, Marion Bowen postdoc grant for the funding source. Thank you very much for listening. I'm happy to take any question you may have.
So we have some questions in the chat box. If you could look at them, we could start with those. If there aren't,
okay.
Yeah. So I'll wait for you to take a second to read it. And if you don't mind repeating the question, Shan, so that we can.
I see. Okay, the first question is about the synchrony study. So this person asked whether if there is a movable glass panel were placed between the two mice in the tube. And I'm curious about how that could relate to humans' online interactions, which is increasingly prevalent during the pandemic and in the digital age in general. Yeah. So in the animal experiments we performed, is like opaque separator between the tube, but the separator cannot like fully block there's like cannot block all of the sensory cue. For example, they may still to like scream to each other, and they can still like smell to each other, but the separator only block their physical interactions. And we found that their synchrony like dramatically goes down. But in human life, as we use so like many different ways to communicate with each other, so for example, this online like interaction, I believe that there are still still some extent of synchrony between the interacting brains, and there are already studies for in the human hyperscanning studies, like when two people like speak to each other, or even they watch a same like movie together, their brain activity are synchronized. So yes, I think for the human online interaction, there will be still significant synchrony between the brain.
And Shan, Felix also wrote a question about male preferring animals. Are they less aggressive and more subordinate, or the other way around? Is there any correlation that can allow you to predict which class a male will fall in, other than the circuit experiments that you talked about, which is really fascinating? Because are these animals isogenic, by the way? Are they, CF7 black 6 taconic or CF7 black 6 Jackson?
Yes, they are just the regular black 6 animal purchased from Jackson library.
So how they become different?
I think this is a great question, but we don't know. I think just individual animal, like probably this encoding is built in their like wired system. It's not, they were raised in the same way, like from they were born. So we don't know what makes them to have a male or female preference, but this has been observed in many other groups also, that individual animal have a different preference for different sexes, although there's an overall preference for the opposite sex at the group level. So it's a great question, but we don't know how that is related. But as Felix asked, whether like it's related to their dominance level, I think this is quite interesting suggestion, and we should look into that after today.
So the other question from An Wu, whether you know anything about the upstream circuitry to the MPFC that you described.
I see. So in this study, we only look at the MPFC like within this region. We also checked some downstream circuit, and we found that the male and female labeling cells, they send projections to multiple brain regions, like in the subcortical area, such as nucleus accumbens, VTA, like striatum, and lateral hypothalamus. But there is, but based on our like tracing studies, we didn't see an obvious difference of those projecting regions. For example, whether females cells send the projection to a very different area. It's not that the case. So they send the projection to similar areas, and there's a not obvious difference between them. For the, like people usually see like sex presentation in the medial amygdala and the hypothalamus. Those centers, they don't have like single synaptic connection to the MPFC. But so we speculate that there should be some intermediate regions that connect the medial amygdala hypothalamus to the MPFC. And those intermediate regions may send the inputs to the MPFC with a more integrated or processed cues already. So yeah, so sorry, we don't have a direct evidence about like whether the male female representation is coming from the upstream regions.
You know, I have just a very brief follow-up to that question.
Mm-hmm.
You know, the medial prefrontal and other prefrontal regions are part of the executive centers in the brain, right? So they are managing information that's elsewhere, and then directing behavior. So how do you reconcile classic roles of medial prefrontal with this role that you've discovered? How do you see the whole system working?
Yeah, so main function for the medial prefrontal cortex is like it integrates all of different information. For example, it integrates the information from others and with information from the environment and their own internal states. So MPFC is a region that integrates all of those information. And because our decision is not solely based on any one of them, it has to be considered the whole whole environment, like whole status. And MPFC is doing such a role. Like it perceive the inputs from all the sensory inputs from other cortical region, and integrates all of the information and make the decision, like what should I do to generate behavior response.
So Joseph Armani is actually a question that's related to what we are just talking about, which has to do, what's the relationship between neurons that respond to male and female cues and sexual attraction, coupling and other behaviors. Have you seen any relationship, between the preferences that you have, described and other behaviors, such as coupling, probability of coupling or attempts to couple with, with other mice, whether they are female or male? Over the years, I've noticed that often males try to couple with juveniles, for example, although they are males too. So, coupling behavior is not necessarily directed towards animals of the opposite sex. So have you seen any relationship between the two? Meaning what you've described, today, and as Joseph Armani puts it in the question, sexual attraction or behaviors that reflect sexual attraction.
In this study, the sex preference, like we define in this study, is simply meaning the male or female directed sociality. Yeah. So it could mean differently from the sexual attraction. One example can be like my best friends are females, but my husband is a male. So the two could mean different things. But in our study, we just simply look at whether the animal like to spend time with male or female. So it may not necessarily mean there is a sexual attraction. But I believe that to measure that, one has to measure some sexual hormones or some sexual behaviors in that case, but it's not what we see today.
Yes, so there is also a question from Xiang Peng about olfactory signals that you mentioned that are important in recognizing behaviors of the opposite sex, and whether you could elaborate more on the role of audiovisual cues in this process.
Yeah, great question. Yeah, so the literature mainly focus on the olfactory cues, or we call it chemosensory cue, those pheromones or odorants, because that's the main pathway that the rodents use to recognize sex. However, the rodents also use visual stimuli or auditory cues to recognize, because the different mice will have different ultrasound wavelengths, not different ultrasound signal. So that depends on the sex. But we don't have much information about how the sex is recognized through those pathways, but it's definitely one important contributions to the sex recognition. But I do have some information about human, how human recognize sex. So people found that the human recognize male or female is mainly through the visual stimuli. And the circuits or the region that's responsible for male or female representation is built within the face recognition network. Like people usually just recognize their face features of male, female. But for the mice studies, it's very hard to measure those. So we do not have more information about other sensory cues.
So now for the last question, which comes from Felix too. He mentions that you talked a little bit about development. And I find this absolutely fascinating. What about juveniles and aged animals? Are there changes? Where do these preferences, when do these preferences emerge? Are they stable throughout a lifetime? Clearly, this is a fascinating study and there are so many questions. That's the signature of a fascinating study, when you suggest more questions than you've answered. So what about the stability of these phenotypes?
Yeah, that's such a cool question. like whether juvenile or age animal also, like when does this sex preference start to show? We don't know. Like I don't know, but if I speculate, then I do believe that is after they become mature, like after they like come into the adulthood. I do have a piece of evidence we tested, but it's not using the juvenile as subject mice, but use juvenile as the intruder. So whether adult subject mice have a preference for male juvenile or female juvenile, and we didn't show we didn't see a preference for male versus female juvenile. So this, although the experiment is done like in the opposite way as Felix asked, but it do have a suggestion that if the mice is very young, like at the juvenile stage, they probably don't have enough sensory cues for the adult mice to perceive as sex information. So so that's a great question. And I also wonder like whether the aged animal have decreased or this sex preference is stable, like when the animal getting old. But in the short time scale, like between days, like day 1 and day 2, their sex preference seems to be stable, like consistent with each other. But we didn't test for the long run, like when they, like several months after. But that's a quite interesting question, and we should look into.
So thank you. This was an absolutely fascinating and incredibly well-given lecture. You are to be congratulated, and thank you. I think this brings this to a closure. If there isn't any pressing questions, if anyone has a question that they're, absolutely have to, to ask, please let us know. If not, we want to wish you a wonderful afternoon, and this was quite a treat. Thank you, Shan. Thank you.
Thank you so much, Alcino, and everyone for listening.
All right. Bye.